In the previous class, we have talked about pollination and their types. We have also discussed about the different mechanisms that a plant devises to achieve pollination. Now we are going to talk about the events that are taking place after pollination. Once the pollen grains, they fall on the stigma. This is the stigma. And the pollen grains, they fall on the stigma. This is the work of the stigma to recognize the right type of pollen grain. The pollen grain has to be compatible, so a stigma continuously interacts with the pollen grains to find out whether the pollen grain is compatible or incompatible. Now this interaction or the dialogue between the stigma and the pollen grain is mediated by some chemical substances which are released by the stigma only. This is known as pollen pistil interaction. This is known as pollen pistil interaction. And it is a major event in the angiosperms. Now after this interaction, once the stigma finds that the pollen grain is compatible, it stimulates the pollen grain to germinate and develop the pollen tube. So a pollen tube will be germinated. Now how does the pollen tube gets germinated? You have already learned that pollen has got an outer cover of exine and intine, a very hard cover. But there are certain points where the intine is very thin or absent. Those points are known as germ pores. From one of the germ pores, the pollen tube will germinate. So a pollen tube finds its way towards the stigma. So this whole phenomena of germination of the pollen tube is known as siphonogamy. This is known as siphonogamy. If one of the pollen tube germinates, then it is known as monosiphonogamy. Monosiphonogamy. If many pollen tubes germinate, then it is known as polysiphonogamy. This is known as polysiphonogamy. No matter how many pollen tubes are germinating, it is only one pollen tube which develops and reaches way towards the micropyle to enter the embryo sac. All of you have seen earlier that pollens are shed on the stigma in two-celled condition. That is, one is vegetative and the other is generative. The vegetative cell gets converted into tube nuclei and the generative cell divides mitotically to form two male gametes. So two male gametes along with the tube nuclei and some portion of the cytoplasm is pushed towards the pollen tube. The pollen tube along with all its contents will now find its way towards the micropylar end. Now if the pollen tube enters the embryo sac towards the micropylar end, then it will be known as sporogamy. If it enters directly from the embryo sac, that is from the chalazal end, then it is known as chalazogamy. if it enters through the integuments, there are two integuments. So if it enters through the integuments, then it is known as mesogamy. The pollen tube, along with its contents, that is two male gametes, cytoplasm and one tube nuclei, has to reach the embryo sac. Now how does it reaches the embryo sac? So let us draw another diagram of the embryo sac. This is the chalazal end and this is the micropylar end. One egg cell is present and two synergids are present at the micropylar end. Then there are antipodal cells. Three antipodal cells are present in the chalazal end. One of the synergid will start disintegrating. So let's see. Synergids have got finger-like projections known as the filiform apparatus. This is filiform apparatus. This is filiform apparatus. The filiform apparatus guides the pollen tube towards the embryo sac. The degenerating synergid will now degenerate and the pollen tube will burst into the embryo sac with all its contents. It is going to put all the contents in the embryo sac. This filiform apparatus along with the synergid will also degenerate. One of the male gamete, in fact the first male gamete, one male gamete which is haploid will now fuse with the egg cell. It will now fuse with the egg cell which is also haploid. So this is going to give rise to zygote which is diploid in nature. This is diploid in nature. Now this whole process is known as fertilization. This is known as fertilization. Fertilization or syngamy. This is known as fertilization or syngamy. Now one more nucleus is present. The male gamete is present. So the second male gamete will now travel towards the center and fuse with two polar nuclei. So second male gamete plus the two polar nuclei. Polar nuclei, they are also haploid. So that means 2N. So this gives rise to primary endospermic nucleus. This giving rise to primary endospermic nucleus. Now this whole process is known as triple fusion. One male gamete fusing with two polar nuclei and giving rise to a triploid condition known as primary endospermic nucleus. So this is triploid in nature. So this is known as triple fusion. Triple fusion. So triple fusion plus syngamy is known as double fertilization, which is a very important characteristic of all the angiospermic plants. So triple fusion helps in the formation of the primary endospermic nucleus, which later on becomes the primary endosperm cell, which again gives rise to endosperm. So triple fusion and syngamy, that is fertilization, together is called as double fertilization. This is known as double fertilization. Double fertilization. Now this was discovered, studied by Navashin in the year 1898. He studied this in two plants, that is lilium and fritillaria. Fritillaria. Now the question comes, which develops first, the embryo or the endosperm? After the formation of zygote, the endosperm starts developing because it has to give nourishment to the growing embryo. The endosperm acts as a reserve food for the growing embryo. Now let us recall again what we have studied. The germination of the pollen grain, that is the development of the pollen tube, and the pollen tube reaching with its content, that is two male gametes and one tube nuclei, along with certain amount of cytoplasm reaching towards the micropyle. This whole process that is still here, it is known as pollen pistil interaction. Now the male gamete, one of the male gamete fuses with the egg cell, which is known as syngamy, and it results into a diploid condition known as zygote. It results in the formation of zygote. The second male gamete will fuse with two polar nuclei and result in the formation of the primary endospermic nucleus, which is triploid. So this was double fertilization, which is very important characteristics in the angiosperm.